All right, everybody. Thank you so much for coming. We really appreciate you stopping by at Reef for our monthly Fish and Friends session. Before we get started, I've got a few community announcements to take care of. So we've got our Fish Out of Water 5K is coming up. We have over 560 people registered, which is crazy. We actually had to order more shirts. So if you haven't got yours yet, don't worry, it's coming. And registration is still open. Go online to reef.org and check it out. We also have Trivia Night at the Florida Keys Brewing Company coming up on 5/26, which is a Thursday, May 26th. Join us at Florida Keys Brewing Company, and we'll be talking about some fun ocean-themed facts as well as just fun facts in general. So study up and bring your trivia knowledge. This summer, we're going to be doing single-day summer camp programs every Wednesday, starting on June 8th and running through August 3rd. So if you know anybody who's interested in doing a single-day summer camp program, know anybody who's got kids, grandkids around and looking for something to do, every Wednesday this summer, starting June 8th, Reef has got something going on. And this isn't on here, but our Lionfish Derby registration is open for fall 2022. The Derby will be on September 9th, 10th, and 11th. On September 11th will be the actual Derby down at Postcard Inn in Islamorada. So we would love to see you join us for a fun day of celebrating removing lionfish from the reefs. With that, does anyone have any community announcements or anything going on? Yes, what you got, Bob?
Next Wednesday, the History of Diving Museum has a new exhibit opening, the Sponge Hunters, that follows up on Linnea's talk. And one of the Greek divers from Torpid Springs is coming in person. But that's a brand new exhibit.
All right, so check out Sponge Hunters next Wednesday.
Hal makes a good point. Not next Wednesday, which would be tomorrow, the Wednesday following.
2 Wednesdays from now. Excellent. And I know that you can, if you're interested in outplanting sponges, to go with that. Key Dives is doing sponge outplanting every Wednesday morning. You can meet Bobby. I can't remember Bobby's last name. I know she goes by Bobby Renfro, and she goes by Sponge Bobby. So that's very exciting. So if you would like to go outplant some sponges, definitely do that. You can sign up through that, I think, through the History of Diving Museum or through Key Dives. All right. And it is free for locals. So even better. And with that, we were so excited to welcome Emily and Loretta and Alexander to talk tonight. We are very excited to welcome Emily back to Reef. Emily has been with Reef since the very beginning. She's Reef member number 9, which is a long time ago. And I will let her talk more about that. And with that, Emily, welcome. Loretta, welcome. We're so glad you're here.
Great, thank you. It's probably connected to there.
Yeah, let's see if I can get it going. Oh, it's moving on the computer.
Oh.
Let's try moving this one over here.
Oh, there we go.
How about this version?
Aha! All right. Well, thank you so much. I turned on the mic. Can you hear me all right online? All right. Thank you all so much. It's a pleasure to be here. I had to bring my favorite hat, of course. Mostly I'm happy because I still have it. It's been 30 years or so. And when I was finished being an intern, somebody gave me this. I'm pretty sure it was Laddie Akins. And managed to have it. I'm really happy about it. So today I wanted to just talk a little bit about community and conservation, and especially the with time that's gone by, I've come to realize that really it's about building community, and that's what's really important, and that sustains us in our efforts and learning, and it really gives great joy. Honestly, right? Because that's why we're all here, I'm sure, is because we love the community that we get. I have to have Loretta, she wants me to do most of the talking, but Loretta made it so that my experience in conservation could really happen. I was out in the middle of the with my work, I had a boat that got taken away. I couldn't use it anymore. And I had lots and lots of herbivory experiments set up, which I'm going to tell you about. I had tiles out on the ocean at Conch and Pickles Reef, and I was trying to count algae when the fish couldn't get there, and it was really vitally important to me. And the Nature Conservancy at the time had a network where people living in the Keys could say that they had their boats available or their time available. And it's a lot like Reef does to help mobilize the community. And that's when I got to know Loretta. So I'm now a professor at Nova Southeastern University. I manage the biology department there. If you want to come to NSU, look around, look me up. I'm in the biology side, but my friends are over there in the ocean campus, and we've got a beautiful campus up there. So I made a word cloud for us. Can you pick it out? Research, courses, education, community. So the name of the game tonight for what I'm going to be talking about is community and conservation, networking, the importance of really getting your team, and the wonderful things we can do when we work together. And I know I'm like telling this to people that already do this, but I'm hoping that something about this story will give you some joy, make you happy, and help us be more connected. So fish surveys, Reef, for me, it was the Nature Conservancy. And I came in to University of Miami, and my professor there, Dr. Kathleen Sullivan Seeley, she was working with the Nature Conservancy at that time. And Ned and Anna, Ned actually, and Paul had gone to the University of Miami and said, who can help us with the data? And the faculty there, Kathleen said, well, I know that there will be students that will want to do this. And sure enough, and we got a computer science person named Ken Marks, and he made the data sheets. And then I show up, and I got to figure out how to get the sheets to the scanner. And I didn't know how to do that for a long time. So they would just pile up in my room. And it was scary because I was starting to get 100 of them. And some of them were on photocopied sheets, if you can believe it. And I was like, I know this is really important. We got to get this stuff digitized. So we did. So this is the team. I got my team to come with me. So lately, we've been doing divers against debris. Have any of you done that? Yeah, that was fun. I met up with the Island Ventures team right when my kids, when I got involved with Reef, I was 21 years old, and I'm, 50 now, so a long time ago. And now my kids just learned to dive. So I got an 18-year-old and a 13-year-old. So that's how we got into Divers Against Debris. And we reconnected back with Loretta, who had the boat and a science background and helped us with research in the past. So that's me. I don't know if either one of you want to say a word. Introduce yourself. So who are you?
All right, I'm Alex, the guy on the right. And Divers Against Debris, it's been really amazing for me because when you're doing the program, you actually get to see a lot of what the conservation efforts are bearing fruit to. You see the number of trash you've picked up. You see the poundage. You see that sometimes there's been days where we've had over 300 pounds of pure garbage from a single dive. And it's just really incredible to see what a relatively small group of people in the grand scheme of things, like 8 or 10 people, can do in a single day to help improve the health of the reefs.
And Loretta's been in the Keys, I guess, for like 40 years, maybe 30.
No, more like, that's a big gap. There's a little private joke here. Loretta Lawrence, I've been in the Keys for 31 years. Okay, and started volunteering, via the Nature Conservancy, which was a fabulous clearinghouse, which they don't have anymore, of opportunities to be able to dive or use boat support. And Emily was my first PhD student, and I had another one after that I did research with. I've done conch research with Bob Glazier. Some of the old timers are here, knew about Bob, right? We did that. We did lobster research. We did algae research, a lot of algae research down at Moat. And this is all as a volunteer. Okay, I just want to tell you. So I learned a lot. And then in the last 20 years, I was taken under the wing by Dr. Judy Lang. If you know about Dr. Judy Lang with AGRA. So I've learned AGRA, I teach AGRA. I was teaching AGRA Light at the high school for almost 20 years, under Dave Makepeace, who actually started the wonderful marine studies program there. So the kids and I would go diving and I would teach them core ID, reef health assessment, and they learned to do percent live coral cover, percent algal cover, and we actually have this data for almost 20 years showing trending in the wrong direction, okay. But it's there. And so that's been interesting because once the kids get to learn what they're looking at, really looking at, because everybody looks at fish, oh, look at that pretty fish. Look at that pretty fish. What kind of fish is that? But most people don't know what coral looks like or what live coral looks like. Okay, they really don't, and then once you start teaching what live coral really is and what ID they are, what kind of predation and bleaching and disease, which we've had a lot of disease recently, it opens their eyes. They don't look at the reef the same way again. And I think that's what's important. Be able to teach people what they're really looking at and the value of why we do this. Okay, that's it. Here you go. That's all you read.
Okay. Well, thank you. So now I'm going to tell a story of how the community saved a lot of research and my little PhD, which I greatly appreciated. So it's a pleasure to be here and talk with you all. Let me see. I was going to try to find my talk over here. Well, I won't do it, but you can, if you ever want to look me up, you can go look up NSU, Nova Southeastern University, Department of Biological Sciences. Emily Schmidt-Lavin comes right up, and that's the department I'm running presently. I'm very proud of it. We have a graduate program. We have master's students. We have a TA scholarship program, and we've really grown it. So I'm happy to talk about that too. I had to look really hard and find some pictures from the past because we didn't take pictures in the past. And if we did, they're physical copies and who knows where they are. But a few years ago for Career Day for my daughter, we made a scrapbook of how we got where we were. And I found this gem from 1995. And that is actually me, and my brother was helping me, and that's Lad Akins over there, and Cheryl Shea, if some of you know her. And that was not the first reef trip, but that's the first one I could find a photo of. So maybe somebody else has one. And then I was fortunate enough, the Nature Conservancy actually got a photographer. I was always trying to say, all right, once I got my work started, how was I going to regularly go back and be able to survey the work that I started? And one time a photographer said, well, if you'll wear my gear and get in the picture, I'll take you to your reef. So that worked out, and it made a really pretty picture. None of the gear that I'm wearing is mine, because I'm telling you, I had the worst gear in the universe when I regularly dove. It was so bad that Quiescence was the only group that would service it. It was really off-label, and I'm thankful they would do that for me. Anyway, now fast forward years down the line, and well, not my brother, it's my son. Loretta's there, my husband, and a colleague, a chemist, and somebody who I did my PhD work with. And there we were that day at Island Ventures, because when we got the kids to learn to dive, we discovered they were doing the Divers Against Debris. So the theme is it's the community and building community, and really if we can build a clearinghouse for people who want to help in the community, and that's Reef does some of that, and other groups do too. It's a wonderful thing. So here's my little story. I hope I can be somewhat entertaining. And I kind of grew up. Reef helped me grow up, you could say. It was my first job out of college. I graduated college and I was 21 years old, super excited to have I was actually going to get paid enough to live a little in my teacher's grandmother's house down the road by the Largo Cargo. And I pretty much had enough money to eat, but I mean, I was a lot. I didn't eat much, but I had enough to survive. And Ned and Paul had reached out to the Nature Conservancy, like I said before, as they were preparing the Reef Environmental Education Foundation to grow and to have the fish surveys. They contacted my advisor there at the university, and Dr. James Bonsack, and so they gave him the hat that has 007 on it. Dr. James Bonsack. Now he's since retired to the Smoky Mountains area, which funny, I aspire to do that too, maybe, but that's where he's at. So 1993, that's where it is. So I'm 21. I come to the PhD program at University of Miami, and I met Christy and Bryce because they were also here in the Keys. We're all going to be interns that summer. And we did, and we had a great time. And, I began the PhD program right then. And then I needed to do a specific project. I did reef surveys, and then I also did this other stuff, the influence of herbivorous fishes on coral reef communities with low sea urchin abundance. So at the time, there was a particularly low sea urchin abundance here. And I got to do a study among reef community types and seasons in the Florida Keys. And this was quite fortunate because my advisor had a NOAA grant right then, and we were going to have some ship time. So that was a little bit of community and, fortunate chances really to get to see a lot of the reefs and document the fish communities that were there. Then 1996, I thought things were pretty good in 1995 because we had research funding from NOAA. We had the Nature Conservancy got donated a 26-foot Mako boat, and I put my little enclosures for getting algae off the reef, which I'll show you what that looked like. And everything was pretty good, but then I had no more boat. We lost access to the boat. We lost the funding for using the boat. I was not allowed to go to the NOAA dock anymore. So, what are you going to do? Well, at the time, you're going to call the Nature Conservancy headquarters there in Big Pine Key, and they had a Key Marathon. They had one in Marathon, and see if there's anybody who can help you. And that's when Loretta was able to help me. And little did I know, she already had a biology background. We were fellow Tri Beta, which is the National Honor Society for Biology. I didn't really learn that until I was writing our bios. But I knew we were kindred spirits and everything was going to be okay. And she even has family members who are PhDs in statistics. So everything was wonderful and happy story. And I stayed employed in the field of biology, which I love. So what did I do? I'm just going to tell you the story a little bit. And I ended up with 383 herbivorous fish surveys in this project, which I didn't ever present it like that, but that's a lot of effort. And I did 50 by 2 meter transects, and I looked at 3 different kinds of reefs. And then I also would take 2 by 2 meter quadrats and get some, even more eager students than me from the University of Miami to go with me and sit there and count the bites that the fish were taking in 10-minute intervals. Yeah, so I call those herbivory, what did I call them on this thing? Bite counts. So what that was, and now my friend John Kelly, he grew up and he likes to study sturgeons on the west coast of California. Well, actually, he was going to become department chair at his university like me, and he just sent me a note and he said, I'm out of here. I'm going to go play with sturgeons in California. In case John's probably not listening. Anyhow, that's what we did. We counted the bites and that bite count, like how many bites are they taking? So the idea was you would get sort of an index of how much scraping or consuming of the benthos was happening. And then then I made these herbivory exclusion experiments, and I'll show you what those were. That ended up with 588 data sheets. I didn't count it that way when I presented it, when I did the work, but now, since I thought really about, let's make a talk about community and science and conservation and bringing people together, that was a huge effort. I'm not really sure I could go back and do that again, because that's like, what, 600, 7, 8, 9, 10, 11, it's like 1,200 data sheets. I still have them in my office. I just can't part with them yet, but they're there. And I would do these 25 point counts on little tiles. I'll show you what it is. So that was just to organize me. We put these exclusion experiments on conch reef in the winter, in the summer, and then at Pickles Reef, because it's a little bit different kind of a habitat. And the point here that I really want to make is we had over 1,000 hours of volunteer or student time in the water, and that was a huge community effort, and things like this go on all the time and makes it fun. So what did I do? I'm going to just explain what I did as the example of a community project, but and it's old data now, but there were 3 different kinds of community types. So relict reef flat, which is what conch reef and Davis ledge and alligator reef are classified as. And then the low relief spur and groove, that was Pickles and Davis and Crocker ridges. Maybe some of you know these. Yeah. And then the high relief spur and groove, and you might say, Emily, why are they all down there and not up there? Well, maybe you know, because I sort of said it. When I first came on, my professor had a NOAA grant, and we were doing the work down there. So lucky for me, I just did that. And then I actually had to do a little study with data that 007 Dr. James Bonsack had about the fish communities in the upper Keys and how they weren't really that much different from the lower Keys, just to show that it was all right. So that's what I did. I went at these different types. High relief spur and groove is shallow with high relief, and the relic reef flat's about the same depth with moderate relief, and the low relief spur and groove is deeper and a little bit deeper and lower relief. And so I did have some pictures, and these are just actually from the day when I was young and brand new at this, of the example high relief spur and groove and the relic reef flat. Now, I'm particularly excited about that fish. Do you know what it is? That was a day at Conch Reef, and you had to have a special permit. I think you still do to work at Conch Reef. Yeah, it's a cornet fish. How many times do you just see a cornet fish? All the time? Well, then what do I? Really? Well, that's awesome, because I never saw them very much. Maybe they're, well, that's cool. You see them at the Benwood? Well, I'd never seen one, and I was delighted to see it that one day there. I also think I kept my head in the sand a ton. So taking data. But anyway, that was one particularly beautiful day on Conch Reef. And then that's more like Pickles, low relief spur and groove. All right. So, so the theme is you couldn't do these things without community and conservation, and we build community all the time in all these ways, like Fish and Friends and many, many other things that we do. So for these, so first off, I just was looking at herbivorous fish, density, size, diversity, and herbivory rate. So these were just pretty much surveys. So it's not that much different. It's not the roving diver survey, but it's a do your 50 foot. We did 50 foot meter transects, 2 meters wide, and we had to also get the size of the fish. So since you're trying to write down estimated size, we just kept it to 14 species of herbivorous fish. So the parrotfish and the surgeonfish families. And then the herbivory rate, just those same different fishes, species, and their counts of their bites. So it was a pretty tremendous effort. I made this slide new because I was just wanting to make a big deal about all that survey work, really, because we did, you know, because I think like, all right, I can't, for like maybe 15 years, I wasn't even getting out on the water. I was building, my family, career, whatever. And just, it seems to me, when you get that work done, it's incredible. Can anyone relate? Yeah. Oh, and it might be blowing too hard to actually do anything, right? So you get everything ready and then forget about it. You can't do anything. So it's remarkable, really. And it took a community. It took a community through, well, when we had the NOAA funding, it took a community of volunteers who would take us to the reef once in a while. It took a community of dive operators once in a while who would take us out and just let us basically bum around, and they could talk about what's that kid doing with the 50-foot meter line, and we were educational in some way, or curiosity. So that's what we did. And so what did I find? Well, and you might say, why study herbivores in the first place? But I kind of think probably you think about herbivores a lot in this audience. But the coral reefs have high levels of primary productivity. There's a lot of algae on the reef. They're helping us with energy transfers from the producers to the consumers. And herbivores regulate the amount of algae on reefs, and especially at the time when we didn't have that many sea urchins to eat the algae, we're pretty happy with the herbivores eating the algae. And we don't have a huge, and I still don't think we have a huge pressure to eat parrotfish so much here in the Florida Keys, especially not like we do in other parts of the world. So if you have a loss of herbivores, then you would tend to have a dominance in benthic algae, a change in species, a change in a community, a change in an ecosystem. And when we think about community in conservation, we have a community on the reef too. All right, so there is some old pictures of fish eating. I thought I changed that slide to my own, but anyway, the sea urchins were at very low abundance during the late 1980s through the 90s when this was being done. And it was, fortunate for me at the time, I could look at this, this system. All right, so what kinds of questions could be answered with this survey effort that we know we're building community? It took a lot more than one person starting their school project to get it done. So what are the population and grazing characteristics of these fishes? What kinds of algae grow in the absence of herbivorous fishes, more or less? So what are the population and grazing characteristics of these guys? And that was with my old CNC. I actually took the old pictures instead of using the new ones, like with film, like goodness. And I'm just so pleased to be able to talk with you all, because there was no Reef Interpretive Center. And it's a joy to see that there is one, and there's plenty of data sheets and lots of members and a nice, wonderful community. And of course, so we got the ones that do the eating. And that is not from here. I know someone's going to call me out on that crinoid.
Crinoid, yeah. Crinoid's a giveaway.
Yeah. Yeah, that crinoid. And I was so proud of that parrotfish picture, but I did give it to one of my teachers in Maryland, to Towson University in Maryland. And that was a summer trip to San Salvador, Bahamas. And I was so proud of that was on film, okay? So I was really proud of that. It was a lot of work. You only got like 24 or 36 chances. I mean, you had to be good. Some of you know better than me. You had to bracket. Like, I was really like, okay, yeah. But my teacher loved that crinoid. He's the invertebrate professor. So it reminds me, everyone in the community is going to see and give us different things there. So anyway, these are the guys that I was studying. Really, I was counting their bites and so on. So do the characteristics vary among reef types? Do the grazing characteristics vary? Do population characteristics vary among seasons? Do these grazing characteristics vary among seasons? So it Yeah, so it gets a lot with the transects, the quadrats. I put in a more recent picture. As I said, I didn't have anything brand name when I actually did the work, funny as it is, and it barely worked all the time. So this is what it looked like from my little thing, and I'm not going to spend a ton of time really on it, but as I would practice on my mom, she's like, oh, so when the fish aren't there, they're probably going to be more algae, right? And when there's more like structure and things like that, there's probably going to be more fish, right? Right. That is absolutely that's right. But I will tell you, I went to great lengths to document that in very many ways. If anybody ever wants to look, it's publicly available in my thesis. I could get one more download. I don't have that many. But anyway, basically that's what we did, and all that effort and that community building, it made a difference. It sure made a difference to me, and I know other people have used the data now, especially because it's old and it's before there was a lot of protections put into the Florida Keys. So anyway, that's what happened here. This is low relief spur and groove, relict reef flat, and high relief spur and groove. And there were more. This is the different sites: Sombrero Reef. No. It was way down there. It was Eastern Dry Rocks and Pelican Shoals. Remember, I would have loved to do the ones up here, but no, it didn't work out today. Then for me, somebody else did it. And then these were the relic reef, like Conch Reef, Davis Ledge, Alligator Reef, and then low relief spur and groove. That's nope. Try again. Davis.
Okay.
Crocker, because I already had CR, and Pickles Reef. And I did this on my old Centris 610. Nobody knows what that is. It was made by the Apple Macintosh company there, like a long time ago. And anyway, and we actually didn't have PowerPoint. My year, I defended, I guess, in the September of '97. We actually took photos of pieces of paper. Can you believe it? Anyhow, and I, you might want to know what are these letters on top, because I actually also forgot, and I will tell you, it's a non-parametric statistical test. So this was a Kruskal-Wallis statistical test, and then some of you might know it, anyway, it's telling you who's more similar to who. So in other words, when these have the same symbols over them, that means they were really repeatable and like each other. But so was that one, was a little bit like that club. And over here, this one was really different, Pickles Reef. I had data that were really different. So there weren't that many parrotfish and surgeonfish in that survey set, which was 24, only 24 surveys. And so on. And then up here, these two were more like each other. And then this one was different yet. So why did Sombrero Reef, did I see so many, or my group see so many parrotfish and surgeonfishes that time? Well, we did. So there it is. But like my mom would always say, yep, where it was more reef structure, yeah, you saw more fish. Yeah. And then the same idea. So you can see like the same kind of pictures like this. And then I want to make sure I have enough time we can interact. But
how did you normalize your seasonal variation?
Yeah, so this was only in the summer.
So
I only did that in the summer from like May. It was like June, July, and August, I believe. And these were only in the summer. And then these were only in the summer also. So the grazing rate among reef types. So this was only in the summer. And then I did a thing with seasons. Whoops, I just did that twice. I was going to take all those letters off, but then I remembered why they were important and how hard they were. So I put them back. Now this is just doing it for season. So this was the part where we went out in warm, and well, we did it in the quarters of the year. And we saw that the density didn't really change, but what's interesting is the size of the fish did change. So the density didn't change. The size did change. So they were a bit smaller in the fall and the winter, and a bit bigger in the spring. And then the biting of the how many bites do you take, which I didn't really count how much they incorporated into their body, but just how many bites they took. They didn't take as many bites in the colder water, which probably also makes some sense. And it clumped nicely by warm and cold water.
Any sense of the standard deviation? You're reporting the average.
Well, these little things are the standard deviations. So there was a bit of variation. So there was a bit of variation. All right. And these were the big grazers. And then to get to this part about what kinds of algae grow when the fish are gone. So this was my advisor, my faculty member really wanted to have something, we want to have something somewhat experimental. So we're like, well, but how can you do something experimental in nature exactly? And so we're going to make these enclosures. And the first enclosure that I made, it had PVC pipe that went up, and it was about a 1 by 1 meter cage, and it was about half a meter off the ground. Now, do you think that's stable?
No, certainly not in storm.
No, and it was very hard to make, highly labor intensive, and we had 3 hurricanes pass nearby, and it all blew to smithereens. That was a bad design. So instead, I made these Centillo tiles, the clay tiles that are really hard and flat, and you can get them easy at Home Depot, pretty cheap. And I got the netting, and you could put netting, plastic netting over it partway, and so the fish could still come in and out, and you could actually enclose them all the way. Were low profile. You could nail them in pretty nicely with very little damage, and they worked out great. And then I was able to do pickles and concrete, and this is when I worked a lot with Loretta. And now do different algae grow at different reef types during the same season and then different seasons. So my committee of faculty wanted to see this for sure, at least in 2 seasons and 2 reef types. All right. And it didn't, and I had a personal desire to be finished within 4 years. So I finished in 4 and a half. Anyway, that's what it looked like. I took a picture out of the thesis. And now what, in hindsight, looking back at this many, many years later, I really can't believe that I had 7 times 3, 21 cages, and I sampled it 6 times, and I got 126 data sheets, and really was able to finish this. And then conch reef, I put out 8, sampled 8 times, and so we had all this effort. And then pickles reef was a little bit different habitat. So that's what we had. And this is actually, I found a printed photo, and that is me at the time, I think. And you must have, so Loretta had to have taken the picture. I know it's me. I think what I'm noticing is I think I had a little slightly bit better gear at that time, maybe. And can you tell that there's a field of cages? Yeah. Yeah, that's what I wanted to show you. And lucky enough, I won't go now because I'm not sure I can get the connection, but a textbook company came. I was always wondering, how am I going to get to this reef? Because I got it set up. One time the textbook company took us out and actually filmed the herbivore work and put it in a textbook as a digital interview. So that was pretty fun. So it's a community of work. So these are the 25-point counts, and I actually go through and I would write what was under the dot. And then at the very end, I collected what grew and sorted it This is how I knew I found the person I would spend my life with, because he agreed to sort the algae. First, he's like, what is that in your refrigerator? And I live with biologists. And in the freezer, we had the mammals from the other girl. And in the refrigerator, we had the, yeah. So anyway, he sorted. I probably wouldn't do it now, but we sorted it, and he helped me. We even got one red algae in the Smithsonian because somebody, the Littlers, thought it was a pretty cool red algae. Anyway, that's what the cages look like, the tiles, and then the semi-cages. That's considered a cage control so that fish could still go in. The whole concept is the netting, and it's pretty heavy plastic netting. Is it interfering with fish behavior? But they were pretty much bare. And then the cage. And you might ask yourself, how do you secure that easily? And the answer we found was nylon anchor mounts. Do you know what those are? Well, my dad knew what those were from being on boats and tying up wires. And it's this little, don't have it on my keychain anymore, but it's a little plastic thing that a zip tie can go through and a nail can go down. And this worked because it's really hard. You can't spend all your time trying to secure something to the reef. A lot of times they have sticky on one side, but these didn't have sticky, but a lot of times they have sticky to keep the wires up. And you run the wires through. And so thankfully, my dad, the computer scientist, knew how to secure a reef tile. He didn't know it, but anyway, and that's what it is. And you can kind of tell. And they were nailed to the reef only in 4 spots. So it was pretty minimal, and it kept them. They didn't blow around. You can kind of tell here maybe that there's a drilled hole and then it's nailed down. All right, so that was the beautiful spot. And now the data, basically, this is Spiridia and Dictyota. All right, so this is benthos inside the cages during the summer at Conch Reef. And that's Laurencia. So Laurencia turns out to be a yummy red algae. I think the parrotfish really love it. Spiridia and Dictyota, they just love to grow everywhere. So in fact, they also grew in the place. And this was based off of the point counts from putting the little stick in the little place. And I learned what Neomerus is and Dasyclatus because they grew in the winter. All right, so this little fuzzy carpet type algae, that's a Dasyclatus. That's how I say it anyhow. There's some Laurencia, beautiful red algae, and I know that they love this because Loretta and I gave ourselves a treat when it was time to take it down. So I was just going to pick it up and bag everything and document it. But what did everybody want to watch? You wanted to watch them feed. So I sacrificed 3 cages and it was super fun. And we opened it up and we just watched, remember? We just watched everybody come in, all the parrotfish, the wrasses. And then the surprise, I know you're going to remember, we picked open the tile from the bottom and guess what was hanging on under the tiles? Brittle star, big hair. Oh, I don't know, what are they called? The black brittle stars that are quite thick. All right, so I'm making this whole thing that they're herbivorous fish. They only eat algae, blah, blah, blah. Guess what they eat? They love those. They love those brittle stars. Have you seen them? Have you seen parrotfish eat them? Well, I'd never seen it before. I'd never seen it since, but it must be like, I don't know, chocolate, peanut butter cups. When they show up, you eat them. Anyhow, these are some of the algae that I just really enjoyed. And we noticed in the winter that the green algae showed up and the green algae didn't show up in the summer. And like my mom would say, yeah, that, well, it's probably colder, this or that. Anyway, yes, it was cool for me to see that. And I enjoyed learning about those algae. That was the day. We even have a picture. We do not have a picture of the sea star getting eaten, but it was something else. And I know, but remember we were there with big garbage bags and we gathered everything up. And for months and months, I had, just tons of algae in the refrigerator. I didn't own a salad spinner for salad. I owned a salad spinner for spinning algae, and no one could use it for anything but algae. Anyway, that was super fun. It wouldn't have been possible without the community of conservation that we had. And that was the benthos inside the cages at Conch Reef during the summer. So yeah, basically the same kind of stuff there. All right, this is a summary to just kind of say what happened in this study. So in this study, the algae without herbivores, that was the cages. Yeah, we had pretty similar algae grow inside cages at both of the places. And we had both, all the reef types had basically the same density of herbivorous fishes. The size of is a little bit lower in the low relief. But the size of the herbivorous fishes was a little bit less in the low relief, and the grazing was a little bit less at the low relief. And by seasons, I tried to make, it went kind of weird here, but I tried to make some picture to show, but the algal growth, it was less in the winter. The density stayed about the same. The picture's not so great here, but the size got a little smaller in the winter, and the grazing for sure cut off at the winter. And what do you know? I also found less algae in the winter. Yeah, well, okay. So how does this apply to conservation? Well, we did get a window on the algae preferred by herbivorous fishes, and it helps us see the changes that can happen in the herbivorous assembly of fish, and it can assist in management efforts. And a whole big community helped with this. And Reef for sure, the Sigma Xi, the volunteer coordinator, which I think is a wonderful job and something we always want to keep doing with Reef and our communities and my university, and just really thousands of hours and lots and lots and lots of people helped. And quiescence let me dive. Oh, remember they fixed my gear, no one would touch it, and so on. So it's just a huge, 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 humongous effort, and that's what I wanted to talk about. And we're still doing it today and having fun. Now I'm not going to go tell about the other one. I guess maybe one day I can come back, or not. But I'll go kind of quick just through it because I want to get to the last little thing. This is Reef Data, and I had used it to show the fish survey data from different places before it was a protected area. And well, I'm going to tell you, when you protect a reef, guess what you see more of? What's your mother say? Exactly what my mother says. Who is an elementary school music teacher. Science is not her background, but that's exactly what we see. I just documented it in a whole lot of, before the spa was there, you didn't see as many, and after the spa was there, you saw more. And I'll just go through that, and if you want to look at it at the end, you can look at it. But that was super fun to do there. Now, one last little thing I'm going to talk about is that now, all these years later, I've made a science outreach organization, and I really encourage all of you in your community, wherever you are, to think, how can you make a difference and how can you mobilize others to do the same? So take your passion for your career, your things that you like, to help mobilize others and try to make a difference. So we made this group, Science Alive, and I think I probably made it to get my students that I was working with to interact with my kids and probably help me not feel guilty as much about working all the time. So we could all work together. And you can check us out. What we most recently did was hydroculture. Yeah, have any of you ever done that? I think Reef could have a beautiful hydroculture garden here. And I'd be happy to help figure that out. And we tried different ways. We bought these PVC tubes. They're like $80 off of Home Depot. But if you have time and energy, like you are young and probably with time on your hands, you could make your own. But I bought them because I didn't have any time or energy. And we grew a garden at the school, but we tried other ways too. So there's Alex in my office trying with these buckets and what's called an ebb and flow system. Here I bought Sigma Xi, a research honor society, gave us some money to try hydroculture. So this is a tower. Did any of you already do this stuff?
I've seen them. Yeah.
This is really beautiful, and well, some, like I know some engineering departments make their own, and they're very, very cool. This was just a bucket system. That was a tube system. Anyway, I think that I got into gardening. I can tell you, like Ned and Paul Human for sure. He now lives in Davie, like near the Nova Southeastern University. He's got a huge garden. So anyway, I think one of these hydroculture gardens would be awesome.
What are you growing there?
I have been growing, what have we been growing? Lettuce, spinach.
Tomatoes.
Oh, the tomatoes are great. I got a strain of tomatoes from the International Space Station just to make it cool. You can go buy them if they're from University of Utah. They were very cool. The kids thought that was awesome. They would eat the tomatoes as fast as they would grow, these little tomatoes.
Sunflowers. We had
Sunflowers.
What else? Basil, right?
Basil. We also know that there's parasites. White fly is a big problem for us. But we did have that. We had real salad. These are viola flowers. They were pretty to have.
There are folks that sell them here in the Florida Keys. It's called Living Towers.
They make them themselves?
Yeah, they sell them. I think it's on Cudjoe or Amron.
Well, they'd be pretty here. It'd be pretty here. Anyway, there we were. Those were those really cool tomatoes. Oh, strawberries. We tried, and I find strawberries are really hard, but maybe some of you can grow strawberries. We did have a few crops. The kids ate them like before, they were tiny, but they ate them before I could even get there. Anyway, we talked about this. We built the community together. We brought teachers and community members, high school kids, middle school kids, elementary school kids, and had a really fun time. And made a poster about it. So the end of the day, I would say it's a win-win when we combine our skills, passions, resources to impact and connect with the community. And I know that this is the group that does it. And if you want to watch my TED Talk, I have a TED Talk subject. So be inspired. I'd be happy to come to your community and participate in your project. All right. So thank you. All right, so now we have discussion, do we? Is that it?
That was really cool.
I forgot. I was supposed to let him talk about the hydroculture project, and I overstepped.
If anybody wants to know about it, I can tell you after.
Yeah. Any questions on that?
He actually had one of those. I don't know if it was the same people at the nautical flea market. Did you see